So today we will be looking at mental health from a neurochemical perspective. We'll be talking about neurons and receptors. The psychiatrist is involved with medication management. And this enterprise of medication management and psychopharmacology really is based on the fundamental science of a neuron, which is the essential unit or the building block of the central nervous system that comprises the brain and the spinal cord. The neuron actually is the building block of the nervous system, period. Its understanding is crucial for knowing how our medications, or rather our psychotropic medications, work. So what exactly is a neuron? The very first thing is that when you look at the brain, every region of the brain comprise populations of neurons that are in interaction or communication with themselves. And a neuron essentially allows for signals to be sent between themselves, between other neurons. A neuron comprises three fundamental parts. There is a dendrite that acts as sort of an ear for the neuron. It receives signals from other neurons. There is a cell body that comprises the nucleus and other organelles that are involved in creating or the building of molecules such as proteins that serve a variety of functions. And there is also the axon. That's one of the most unique features of a neuron. It's this elongated structure that allows for the transportation of those molecules that have been built towards the axon terminal, the end of the axon. Axons also allow for action potentials to be propagated. So action potentials are like electrical signals that would ultimately allow for the neurotransmitters to be released and facilitates communication with other neuronal cells. So at the axon terminals, molecules are stored in synaptic vesicles. And then an action potential, the electrical signal, then travels down the axon and causes calcium channels to open, and there's an influx, or there is a movement of calcium into the neuron, the neuronal space. And this causes that synaptic vesicle to fuse with the membrane of the neuron. Now, this is the presynaptic neuron. This causes the synaptic vesicle to fuse with the membrane of the neuron, and a process called exocytosis occurs where the neurotransmitters are released into the synapse. So the synapse is basically the space between two neurons. And the neuron prior to the synapse is called a presynaptic neuron. The neuron after the synapse is called a postsynaptic neuron. The neurotransmitters then bind to receptors on the postsynaptic neuron. And two things could happen. If it's positive ion, such as sodium, there is the postsynaptic neuron is activated, a process called depolarization, also called excitatory postsynaptic potential. But essentially the postsynaptic neuron fires an action potential. If it's a negative ion, such as chloride, the postsynaptic neuron is deactivated, or it's hyperpolarized. Process is also known as an inhibitory postsynaptic potential. Let us spend some time talking about this synaptic space and this interaction between the presynaptic neuron and the postsynaptic neuron, because it's very important in understanding psychopharmacology. So like we mentioned, there's the synaptic vesicles that comprises neurotransmitters. There's a synaptic space, and also there could be receptors on the presynaptic neuron. For instance, there could be a transporter, and what that does is that it actually helps remove neurotransmitters from the synaptic space. There are also other enzymes, which I don't show here, that help with the breakdown of neurotransmitters, either in the synapses or in the presynaptic neuron. And this removal process is important in order to terminate the activity of neurotransmitters. So The activity of neurotransmitters could be stopped or terminated in various ways, such as its removal by the transporter or the breakdown of the neurotransmitters by various enzymes in the synapses and in the presynaptic neuron. There's one more process that's very important to understand when we think about how our medications work. And that's what I would call like a basic law in neuroscience. So if you recall from physics, one of Isaac Newton's laws, that for every action there's an equal and opposite reaction, there's something similar in neuroscience where there is a dynamic interaction between neurotransmitters and the receptors. Let me illustrate this point. So you have your postsynaptic neuron, you have your receptor, and then you have neurotransmitters. If for some reason the activity of the neurotransmitter goes down, there is usually a reaction by the receptors where they increase their sensitivity by up-regulating their numbers. So when the neurotransmitter activity goes down, the receptor activity, or rather the receptors, are up-regulated. And if the neurotransmitter activity had gone up, the receptors will be down-regulated. This dynamic interaction, we believe, underlies some psychopathological processes like in depression. We could see how, we could view mental disorders from a neurochemical perspective and define mental disorders as alterations in the dynamic activity of neurotransmitters and their receptors.